Hey everyone, Dr.Mungli here. So, in my previous video, I have explained integration of metabolic pathways under fed condition. So, if you haven't watched that video, so the link for that video I have given in the description below. So, please watch it once you finish this video, so that you have covered integration of metabolic pathways in the fed condition, and in today's video I am explaining integration of metabolic pathways under fasting condition and starvation. So, before we get into the details, so let me explain you certain terminologies here. So, there are some terminologies like absorptive state, post-absorptive state, fasting state and starvation. Let us get into that. So, consider that we have taken breakfast at 8 in the morning. So, and then for next 4 hours what goes on in our body, it is generally referred as absorptive condition. So, I can say if we have taken lunch at noon like 12, so that will be say that is 4 hours, and then once you take lunch you get into again absorptive state another 4 hours. And then after 4 hours we get into post absorptive state. So, which will go on for around 6 hours from that point. So, before we finish that post absorptive state, so we take dinner, right, and again we go into absorptive state there. So, absorptive state for 4 hours and then the post absorptive state for 6 hours. So, total there are like 10 hours, and then after 10 hours we go into fasting state. So, so probably that will be around like 5 in the morning or 6 in the morning or something, and we are going to break that fasting by taking breakfast. So, that is why we call breakfast there, because we are breaking the fast. So, the fasting actually starts 10 hours after our last meal. So, let us look into little more details on that. So, here is the absorptive state. So, which is 4 hours after meals is referred as absorptive state. And then we have post absorptive state. So, this post absorptive state is referred as it is next 6 hours from the end of absorptive state. So, that is 6 hours, post absorptive state is 6 hours, and then we go into fasting state. So, in the fasting state basically it starts 10 hours from our last meal and it lasts until 24 hours. So, from 10 hours after meals until 24 hours, generally we refer that as a fasting state. So, what happens after 24 hours? So, what if the person do not take food even after 24 hours? So, that is generally referred as prolonged fasting state. So, that is it will go from 24 hours after meals until 72 hours. Now, what if the person do not take food, even continued not to take food even after 72 hours, that is 3 days, right? So, at that time, so that particular state we call it as a starvation state. So, remember, so this is what is the starvation. So, there is a confusion here in the terminologies, like especially for starvation, when exactly starvation begins. There is no clear cut like no in the books. Not every book gives the same timeline, but this is I have taken from Mark's textbook of biochemistry. So, starvation state starts from 72 hours, that is 3rd day onwards, and and it goes as long as person is not taking any food, that is in starvation. So, these are the timelines that we have. So, now let us move into metabolic changes during post absorptive condition and fasting and starvation, all these things put together. So, I am going to explain you certain changes that are going on in our blood, right. So, in order to understand that, let us understand the plot here. Initially, I have given post absorptive and fasting state in hours, right. So, 12 hours and going into 24 hours, and then I break this into days, like this is 1 day and 2, 3, 4, 4. So, that is up to 1 week, that is 7 day here. So, prolonged fasting state and starvation, that is the period here. And then if the fasting, if a person do not take anything, so if continue to like not having any food, they basically get into starvation, and starvation I have given in weeks like 2 week, 3 week, 4 week and 5 weeks of starvation. This is how the x axis is divided here, and the y axis plasma levels of molecules are given I have in millimoles. On the right hand side I have given in milligram per deciliter. Okay. So, as the person goes into fasting condition, so from say post absorptive state and fasting state, so that means 6 hours after meals and all the way to 24 hours. So, what happens? So, to maintain the blood glucose level, so the glycogen present in the liver is degraded first. So, the glycogenolysis, it peaks by 12 hours, make sure you remember that, it peaks by 12 hours and then it is completely depleted by 24 hours. This is the glycogenolysis curve, and do not scale this glycogenolysis curve with the plasma levels, that is not for the here, that plasma level is for something else, I am going to explain that soon. So, the glycogenolysis, it starts say 2 to 4 hours after meals, slowly it starts and then peaks by 12 hours and glycogen is completely depleted by 24 hours. Now, no glycogen is left to maintain the blood glucose level. So, what happens at that time? So, the gluconeogenesis begins like 2 to 4 hours after meals, like slowly it begins and then it starts to steam up. So, as you can see, by 24 hours your gluconeogenesis process is already at peak. By that time your glycogenolysis is already depleted. So, gluconeogenesis follows then almost at peak for another 1 week up to like 6-7 days, and then it drops to a certain level and continues thereafter. This is what is the gluconeogenesis curve. We are going to look into the enzymes and also the substrates for gluconeogenesis. Now, let us move on to see what about the blood glucose level. Now, the glucose levels in the blood, say when glycogenolysis, glycogen breakdown is going on. So, blood glucose is maintained and then and that is continued by gluconeogenesis, where the blood glucose is maintained, and as the gluconeogenesis falls here, like after 1 week, as the gluconeogenesis falls to certain level, even the blood glucose level also reaches to certain level there and continue to maintain between 70 to 80 milligrams per deciliter. That is a fasting blood glucose range, 70 to 80 milligrams per deciliter. So, it maintains around like 70 or 80 milligram per deciliter, and that is how the gluconeogenesis will make sure that blood glucose is continued to be maintained for the necessary blood glucose, which means the glucose that is necessary for certain cells and organelles. Okay. So, then what happens? So, the fatty acid oxidation continue to go on as the person is in fasting condition. So, the fatty acid lipolysis is going on, fatty acid flooding into the blood, taken up by the liver, taken up by the other tissues. As you can see, fatty acid continue to rise as the person goes into fasting and starvation. As the fatty acids continue to rise, see here like ketone bodies rises in the blood, right. As see initially there is a basal level of ketone body synthesis, significant rise of ketone bodies appear like around 3rd day of fasting, and then continue to rise by 2nd to 3rd week of starvation, ketone bodies almost at peak here. So, this is what these are the plots for, these are the curves for glucose, fatty acids and ketone bodies. Now, let us move on to see what all the metabolic changes that are going on in post-absorptive state and fasting condition. Alright, so as we get into post-absorptive state and fasting state, so blood glucose level as it is falling down, because blood glucose as it falls, release of insulin from beta cells will decrease and release of glucagon from alpha cells of pancreas will increase. So, what this does? So, the liver which has stored glycogen in the liver. So, there are around like 150 to 200 grams of glycogen which is stored in the liver. Now, the glycogen breakdown will start. As you can see, glycogen breaks into glucose. This is mediated by glucagon. Glucagon is going to stimulate an enzyme called glycogen phosphorylase enzyme, and this glycogen phosphorylase enzyme releases glucose from glycogen. So, what will happen to this glucose levels which is coming from glycogen? So, the glucose, it is going out of the liver, so it will be secreted out of the liver into the blood. So, that is what is maintaining the glucose levels in the blood under post-absorptive and fasting state. So, which are the organelles that takes this glucose coming from the liver? So, the most importantly, this glucose is taken up by the brain and also it is taken up by the red blood cell. Remember, brain is dependent on the glucose initially for its energy needs, and red blood cell always it is dependent on the glucose for its energy needs. So, what the brain does? So, it is going to break glucose into acetyl CoA and further into carbon dioxide and make ATPase. Similarly, the like red blood cell, it is going to break, it conducts anaerobic glycolysis, converts glucose into 2-pyruvate and 2-lactate molecule and get those 2 ATPs. So, meanwhile, what will happen because there is a insulin glucagon is increased. So, what this glucagon does, it is going to break the triacylglycerol that is stored in the adipose tissue by stimulating hormone sensitive lipase enzyme, and thereby when the tags are broken down. So, you are going to get fatty acids and glycerol, right. This is the role done by hormone sensitive lipase. So, what will happen to the fatty acids? So, like fatty acids are used by the peripheral tissues including the skeletal muscle and the heart, and also fatty acids will go into the liver. So, let us look into that. So, the fatty acids are consumed by the muscle for its energy needs. So, fatty acids are oxidized, beta oxidation is going on. In the skeletal muscle, like insulin is not there, means insulin is less under fasting conditions. So, they basically carnitine shuttle mechanism is going on, and fatty acids are undergoing beta oxidation to make acetyl CoA, and which are used for energy purpose. Now, what will happen to the skeletal muscle proteins under fasting, post-absorptive and fasting state? So, the initially proteins are not broken down in the skeletal muscle. So, they do not undergo degradation initially in the initial hours of post-absorptive state and fasting. So, the amino acids are basically coming from transamination reactions, and those amino acids they go out of the skeletal muscle mainly in the form of alanine, and the next amino acid that goes out of the skeletal muscle predominantly is the glutamine. So, the alanine and glutamine get out of the skeletal muscle into the blood, and once they get into the blood, taken up by the liver, and in the liver the carbon part of amino acid alanine and glutamine is converted to glucose, whereas the nitrogen part of your amino acid is converted to urea, and the urea is secreted out of the liver. It is taken up by the kidney, and kidney is going to excrete that urea in the urine. That means, whenever gluconeogenesis is going on, expect that urea synthesis is also going on in the liver, because if you are considering 2 alanine, so you need 2 alanine to make 1 glucose molecule. So, when you have 2 alanine, you have 2 nitrogen there coming from the amino acid amino nitrogen. Those 2 nitrogen will be incorporated to make urea, which is NH2CO NH2, and then it is secreted out of our body in the urine. So, that is what happens. Now, what are the other things that can make glucose, so that which is participating in gluconeogenesis? It is the lactate coming from anaerobic glycolysis taken up by the liver and converted to glucose, and also the glycerol coming from adipose tissue is also taken up by the liver and is converted to glucose. That means, all the enzymes that are necessary for maintaining the gluconeogenesis process here, it is all kept active by glucagon. So, glucagon is to it will keep all the necessary enzymes for gluconeogenesis process in an active condition. Now, let us see what will happen to the fatty acids in the liver. Fatty acids get into the liver and undergo oxidation, that is the beta oxidation, which is again promoted by decrease in the glucagon decrease in insulin and increase in glucagon level, and ATPs are coming up from that. Acetyl CoA can be used for ATPs, and some of the acetyl CoAs are diverted into ketone bodies. Now, note that we are still in post-absorptive state and fasting state. That means, not many ketone bodies are made now right now. So, but the process has started already. So, ketone bodies synthesis starts, and these ketone bodies slowly appear into the circulation, and some of these ketone bodies can be started to used by skeletal muscles. Skeletal muscles can start using these ketone bodies. So, this is what happens under post-absorptive state and fasting state. So, let us recap. Glycogen degradation peaks at 12 hours and depletes by 24 hours. It provides glucose needs of red blood cells, brain and neuronal tissues. Gluconeogenesis peaks by 24 hours, and it is at peak, almost at peak for another 1 week, and then drops to maintain at that level to continue after to provide glucose for red blood cells, brain and other neuronal tissues. Substrates for gluconeogenesis at this time, it can be alanine from muscle, glycerol from adipose tissue, lactate from anaerobic glycolysis. Fatty acids from the adipose tissue rise slowly in the blood, undergo oxidation, muscles start using fatty acids for energy, liver start diverting acetyl-CoA into ketone bodies. Okay. This is what happens under post-absorptive and fasting condition. Now, let us move on to see what all the metabolic changes that happens under prolonged fasting condition. You remember, prolonged fasting condition means it will go from 24 hours after the last meal to 72 hours, that is 1 day to 3 day, like 24 hours after to 72 hours. So, what will happen? Again, so the blood glucose levels are low, insulin is decreased, glucagon is increased. So, that means glucose has to be maintained. Note that it is already more than 24 hours, you do not find any glycogen there. So, glycogen is not available here. So, the glycogen stores are not available. So, that means glucose has to come from other sources, it can come from gluconeogenesis process. Let us look into that. So, as you can see in the skeletal muscle, so skeletal muscle proteins start to degrade at this time. As the skeletal muscle degradation, protein degradation goes on, so all amino acids, especially around all these 20 amino acids, out of these 20 amino acids, 18 of them can be converted to glucose, right. So, the amino acids predominantly come out of the skeletal muscle as alanine and glutamine, they get into the liver, and the carbon part of the amino acids, 18 amino acids converted to glucose, nitrogen is converted to urea. So, urea synthesis increased and it is excreted out of our body through kidney into the urine. That is why you can see the line, there is a thick line here. So, the protein degradation has already started under prolonged fasting condition to provide glucose needs of the brain and red blood cell. What will happen to the adipose tissue? Adipose tissue continue to undergo triacylglycerol degradation because hormone sensitive lipase is active. That means glycerol is going out and fatty acids are going out, flooding into the blood. These fatty acids are taken up by the skeletal muscle, taken up by the peripheral tissues for the energy needs, except brain and the red blood cell. Note that these organs, the cells do not use the fatty acids. Now, fatty acids, the lactate in anaerobic glycolysis can contribute blood glucose level at this time, glycerol contribute to blood glucose level. So, the prolonged fasting condition, amino acids coming from proteins, glycerol coming from adipose tissue, lactate coming from the red blood cell, they all contribute to blood glucose level in gluconeogenesis. Fatty acids will undergo oxidation in the liver, as I explained before, and used for energy purpose. By this time TCA cycle is already kind of saturated. So, the acetyl-CoAs are more diverted into ketone bodies now. So, ketone bodies start to appear in the blood, rising in significant quantities now because it is already more than 24 hours of fasting going into 72 hours. So, these ketone bodies are used by the skeletal muscle and other tissues like heart and other tissues, but right now the ketone body used by the brain may be less significant, that is why I have put a thin line, dotted line here. So, it is starting to use ketone bodies, so it start from like 24 hours after fasting and go all the way until the 3 days of fasting, where it will significantly start using ketone bodies. Let us recap this process like prolonged fasting condition. So, this extends from 24 hours after meals until 72 hours, and then glycogen is totally depleted by this time and do not provide glucose for blood glucose. Gluconeogenesis is at peak, provide glucose for red blood cells, brain and neuronal tissues. Substrate for gluconeogenesis at this time is mainly coming from amino acids after skeletal muscle degradation, alanine coming from the skeletal muscle degradation is increased now, glycerol is contributing to gluconeogenesis coming from adipose tissue, lactate is contributing to gluconeogenesis. Now, the fatty acids from adipose tissue is rising in the blood, undergo oxidation, ketone bodies start appearing in the blood significantly, muscles will use both fatty acids and ketone bodies, brains start to use ketone bodies along with the glucose. Now, let us move on to see starvation. What are all the metabolic changes that we have under starvation state? Now, the starvation, as I told you, it starts 3rd day after the last meal. So, what happens? Again, as I have explained before, so the blood glucose level is down, insulin is decreased, glucagon is increased, and that is going to promote all the processes that are necessary for energy needs of our tissues. like glucose is coming from gluconeogenesis in the liver, it is taken up by the brain, it is taken up by the red blood cell for their energy needs. Adipose tissue continue to undergo degradation by hormone sensitive lipase releasing glycerol and fatty acids, and fatty acids are taken up by the skeletal muscle for their energy needs. Protein degradation, it will continue to go on until like 1 week of fasting, like 4 to 5 days, 6 days. So, where the protein degradation is going on. By that time, after around 1 week of starvation, around 50% of the skeletal muscle protein is already degraded. During that time, protein degradation in the skeletal muscle decreases and it will kind of stops. Why? Because if we continue to degrade proteins, there is no point in survival mode here. So, the protein degradation slows down from the skeletal muscle. That means amino acids coming out of the skeletal muscle slows down. Amino acid as their contribution to gluconeogenesis decreases, thereby even the urea synthesis also decreases. So, what is the process that continued gluconeogenesis now? Remember that like after 1 week gluconeogenesis curve falls down to the certain range, that is only because skeletal muscle degradation has decreased. Now, the gluconeogenesis is supported by the lactate coming from anaerobic glycolysis, it is supported by glycerol coming from adipose tissue. So, these are the two things that will continue gluconeogenesis for the glucose needs of the red blood cell, because as you can see glucose needs of the brain is kind of decreased here because brain has started to use ketone bodies by this time. So, the glucose that is coming from gluconeogenesis after 1 week, because it is already decreased, kind of going to 70 milligram per deciliter of blood, that is used by this red blood cell predominantly, because brain has spared that glucose for the red blood cell. So, at this time ketone body synthesis will increase in the liver. So, liver makes more and more ketone bodies, they flood into the blood, and from the blood. So, the ketone bodies are of course like taken up by the brain. Brain significantly has adapted to use ketone bodies. That is why it is sparing glucose for the red blood cell by consuming more and more ketone body for its energy needs. And during this time, as the brain adapt itself to use ketone bodies, like as it goes, as the starvation go from 2 to 3 weeks of starvation, skeletal muscle will decrease its use of ketone bodies for an energy purpose. It will depend more likely on fatty acids rather than ketone bodies, because skeletal muscle spare those ketone bodies for the brain, because brain is sparing glucose for the red blood cell. This kind of metabolic adaptation is going on in our body under starvation. So, these are some of the metabolic pathways that goes on in our body during fasting condition and starvation. Let us recap starvation story here. This starts 3rd day onwards and continue until food is consumed. Gluconeogenesis drops by the end of a week. Muscle protein breakdown decreases, continue to provide glucose for red blood cell, brain and neuronal tissues. Substrate for gluconeogenesis now, it is glycerol coming from adipose tissue, lactate coming from anaerobic glycolysis. Fatty acids from adipose tissue continue to rise in the blood and undergo oxidation. Ketone bodies continue to rise in the blood significantly, and muscles will use both fatty acids and ketone bodies until the end of 2 weeks of starvation, and then decrease the use of ketone bodies and spare them for the brain. Now, brain completely adapts itself to use ketone bodies as the energy source and spare glucose for the red blood cell. So, these are the metabolic adaptations that are going on in our body under post-absorptive condition, fasting state, prolonged fasting state and starvation. Now, let us move on to see what are the roles of other hormones in during this time. Like what is the role of epinephrine and cortisol in the integration of metabolism? Let us be brief in that. So, epinephrine increases release of glucagon from alpha cells of pancreas and decreases insulin release from beta cells of pancreas. It increases degradation of glycogen in the liver and skeletal muscle, increases lipolysis by degrading triacylglycerol from adipose tissue by keeping hormone sensitive lipase active. It increases gluconeogenesis in the liver. Coming to the cortisol, cortisol increases protein degradation and decreases protein synthesis in the muscle. Amino acids are coming out of the skeletal muscle and they will go into gluconeogenesis process. So, it decreases utilization of glucose in the muscle and adipose tissue, kind of creates like insulin resistance type there. Increases lipolysis by degrading triacylglycerol from the adipose tissue. It increases gluconeogenesis, but more interestingly note that cortisol actually it keeps glycogen synthase enzyme active and it increases glycogen synthesis, and this glycogen will be degraded by epinephrine. So, cortisol actually increases glycogen in the liver by keeping glycogen synthase active. Epinephrine is going to break that glycogen into glucose. So, this is what are the roles of epinephrine and cortisol under post-absorptive condition, fasting condition, prolonged fasting and starvation. Now, let us move on to see important enzymes that are active and inactive during fasting condition and starvation. So, let me explain you first the active enzymes under fasting condition and starvation. So, glycogen phosphorylase, as you can expect, glycogen degradation enzyme is obviously active. These are the enzymes activated by glucagon. We have glucose 6 phosphatase, fructose 1,6 bisphosphatase, phosphoenol pyruvate carboxykinase, pyruvate carboxylase. These are all gluconeogenic enzymes which are kept active by kept active by glucagon. Glycerol 3 phosphate dehydrogenase, glycerol kinase, which is helping in the conversion of glycerol into glucose, kept active by glucagon here. Hormone sensitive lipase, degrading triacylglycerol in the adipose tissue, kept active by glucagon. Epinephrine, cortisol. Mitochondrial HMG-CoA synthase, helping in ketone body synthesis, kept active by glucagon. Carbamyl phosphate synthetase 1 enzyme, which is involved in urea formation, is active now because substrate availability is more. N-acetyl glutamate synthase enzyme, which is making carbamyl N-acetyl glutamate, which is the obligate activator of carbamyl phosphate synthetase 1 enzyme. These two enzymes are involved in urea synthesis, those are also active now. Inactive enzymes, glycogen synthase, all glycolytic enzymes, regulatory enzymes, glucokinase, PFK1, PFK2 enzyme, pyruvate kinase, pyruvate dehydrogenase complex, citrate lyase, malic enzyme, acetyl CoA carboxylase, fatty acid synthase, HMG CoA reductase, glucose 6 phosphate dehydrogenase, glycerol 3 phosphate acyl transferase and lipoprotein lipase. All these enzymes are all kept active by insulin. Now, the insulin is low, glucagon is more. So, these enzymes are inactive now. So, all the catabolic enzymes are active and anabolic enzymes are inactive. So, because catabolic enzymes are activated by glucagon, anabolic enzymes are activated by insulin. Now, it is hormonal levels are reversed now, glucagon is more, insulin is less. That is why all catabolic enzymes are active now and anabolic enzymes are inactive. So, like of course, like ketone body synthesis is an anabolism, but that is in the starvation state because it is anyway catabolized later in the peripheral tissues. Alright, now let us look into all these metabolic changes again in terms of plots. Now, we know post-absorptive and fasting state, we know what is prolonged fasting and starvation, and we know what is starvation now. So, the glycogen degradation is at peak at 12 hours, depleted by 24 hours, gluconeogenesis peaks by 24 hours, be there for around 1 week and then falls to certain level and continue thereafter. The glucose levels are maintained by glycogen degradation and gluconeogenesis, and the glucose levels maintained around like 70 to 80 milligram per deciliter for the needs of brain and red blood cells. And fatty acids continue to rise from 2 to 4 hours after fasting all the way until person is in survival mode, and as the fatty acids are availability more, so ketone body synthesis significantly rise and they will be reaching at peak by 2 to 3 weeks of starvation. Alright. So, here is the tabular form, glycogenolysis peaks by 12 hours, depleted by 24 hours, gluconeogenesis peaks by 24 hours, continue almost at peak for around 5 to 7 days and then drop to continue at basal level there. Blood glucose initially maintained by glycogenolysis and then by gluconeogenesis maintained at fasting range. Ketone bodies coming from fatty acid oxidation supports this ketone body ketogenesis. Ketone bodies appear after 24 hours significantly by 3 days and peaks by 2 to 3 weeks. Alright, so this is what is all about integration of metabolic pathways under fasting condition, prolonged fasting condition and starvation. So in my next video, I am going to explain you the regulation of all these metabolic pathways, regulation of carbohydrate metabolism under fed condition, fasting condition, regulation of fatty acid metabolism under fed and fasting condition, and regulation of amino acid metabolism under fed and fasting condition. So my next week's video, it will be on the regulation of these metabolic pathways. So I hope this video has helped you in understanding integration of metabolic pathways. So kindly watch my integration of metabolic pathways under fed condition. The link is there in the below video, and also it is coming up now at the end of this video. And this video is on integration of metabolic pathways under fasting state and starvation. I hope these videos are helping you in understanding these seemingly difficult topics. If you have any question, please write that question in the comment section below. I will see you in my next video. Till then, you take care. Thank you.